So today we're going to be talking about catalysis and how enzymes can lower the activation energy of chemical reactions. So proteins can do lots of things. They can do defense and protection, like for instance, keratin is a barrier against things getting into your body, and antibodies fight off illnesses. Movement within and of the cells. So movement within the cells, the motor proteins, actin, kinesin, dynein, and movement of the cells like actin polymerization and depolymerization. They can do signaling and communications, like for instance, insulin and glycogen are signaling molecules that are proteinaceous. They can give structural support, like for instance, the microtubules. They can transport things across the membrane, like ion channels, uh, the storage of nutrients. There are lots of things that proteins can do, but mostly what we think of when we think of proteins is enzymes, and enzymes are catalysts. Catalysts of reactions. They lower the activation energy and thereby let the chemical reaction uh, proceed at a quicker rate. So, what is a catalyst and why do we need catalysts? So, catalysts are things that speed up reactions, and we need them for life to occur because without an enzyme, the reaction rate is going to be too slow for life to exist. With an enzyme, we have a quick increase in the rate of the reaction up to a maximal rate where all of the active sites of the enzyme are taken up by substrate. So, how is this important? Well, if we think about the average random molecular kinetic energy, some things are going to be higher and some things are going to be lower. The um, molecules that have enough energy to let the reaction proceed is going to be a very small subset of the number of molecules. And so without something that is lowering the activation energy, we are not going to be able to have very many substrate molecules interacting or or undergoing the reaction. And so the way that catalysts work is stepwise. There's the initiation step where the substrates bind to the active site of the enzyme. Then the transition state, which is where the interaction of the substrates happens inside of the active site. So the enzyme substrate complex will undergo a conformational change, changing the shape of the substrate and putting it in a conformation that more closely resembles the transition state. And then the termination of the reaction is when the products leave the active site. So what limits the rate of catalysis is how many active sites you have available for binding. So what is an active site? It is a site that is specifically shaped such that the substrate is going to bind with it. So the substrate has different chemical structure, and that chemical structure may have charges or a physical structure that is going to fit neatly inside of the active site. And when that happens, there is going to be a change of shape of the active site. This is called induced fit. This change of shape actually physically bends the substrate to a conformation that more closely resembles the transition state. There are also going to be coordinating amino acids within the active site that are going to undergo temporary covalent bonding with the substrate to allow the electrons to move around inside of the substrate to facilitate the reaction. Um, so the coordinating amino acids inside of the active site do not have to all be right next to each other on the gene or on the protein. The coordinating amino acids can be far flung within the primary structure, but because of the three-dimensional shape of the protein, they are all going to be right next to each other in that active site. So I commend this YouTube video to you. Watch it, and you'll see what I'm talking about there. So um, in general, the enzyme is not consumed in the reaction. This fits the definition of catalyst. Everybody who took chemistry probably remembers catalysts like zinc or copper or something like that. And the zinc or copper or whatever the catalyst was not used up in the reaction. And so for this to be true, the product enzyme complex has to have very little affinity so that the product will leave the active site. So the covalent bonds that form when the substrate enters the active site have to be temporary covalent bonds. Usually, the reaction is controlled by some sort of feedback mechanism, and so there are generally metabolic pathways. Metabolic pathways where the product of enzyme 1 um, is the substrate for enzyme 2, and the product of enzyme 4 is the substrate for enzyme 5. And very often, the last thing made by a metabolic pathway, in this case, molecule F, will somehow inhibit enzyme 1 so that you'll have some sort of feedback mechanism to control the rate of production of all of the molecules in the pathway. Um, so pathways are regulated. Um, they usually have an optimal temperature, optimal pH. Sometimes other small molecules or coenzymes can also be regulators. Very often, the downstream product is also a regulator. So this concludes the first video of enzymes. Move on to enzyme 2.